So good afternoon, everyone, and thanks very much. The organizer of 3D Genomic invited me, and I'm a PhD student in Akis Papatoni's lab from Göttingen. And today I want to talk about RNA polymerase II and genome folding. Genome folding is a very hot topic nowadays, and scientists have already studied a lot about the mechanisms of genome folding, the relationships between the structure and function. We have already know a lot, but still far away from knowing what is going on. So substantial outstanding progress has been done in recent dozen years, and we have known a lot about genome folding, but we still far away from knowing what is going on. And a short introduction of what we have known about genome folding is, firstly, in the nucleus, each chromosome occupy their own territory rather than mixing with each other. And if you look within one chromosome, you can see it is divided into megabase large domains, mostly active or mostly inactive. And to some extent, they segregate. And people named this active region as compartment A and this inactive region as compartment B. If you go through with a high resolution to these hundreds of kilobase structures, you can find a kind of dominant structure called loops. And there are usually, in general, there are thousands of conversed and stable loops in human genome, and they are formed by this cohesion looping process. And cohesion is a ring-shaped protein that can load it on chromatin via RNAPll. And it can also, when it's loading on chromatin, it starts extruding chromatin, looping chromatin. Until cohesion meets a convergent element, CDCF, it stops extruding and to form stable CDCF-based cohesion loop. And in general, about 20 minutes, cohesion is removed from chromatin via this WAPL and PDS5. So because nowadays most of the research is focused on this CDCF-based cohesion looping model. But cohesion is not the only protein that can translocate DNA. So we're curious what is going on when cohesion looping process meets another chromatin translocation process, or just what is going on when cohesion looping process meets a barrier, a protein that is binding on chromatin. So in our project, we think RNA polymerase II is such an important protein that binding on chromatin and translocating chromatin for a long time during G1 phase to fulfill transcription. So we're curious what is going on when cohesion meets RNA polymerase II. And also there is a paper in bacteria. It is say RNA polymerase can act as barriers to a bacterial condensin loop extrusion. So we're curious what about human genome? And secondly, RNA polymerase II is one of the bridges that can link structure and function to answer the question, what is the relationship between structure and function? So to answer these questions, we received as a gift a human cancer cell line, DLD1, from Mishima Lab in Japan. And it can inducibly and reversibly degrade the largest subunit of RNA polymerase II, that is RPB1, by adding auxin. Nadine Benmansa did the experiment. We checked the degradation by Weissen blot and immunofluorescence. So here you can see after 14 hours, 90% RNA polymerase II are gone. And the degradation of RNA polymerase II didn't affect other RNA polymerase 1 and 3. And here you can also see the decreased chromatin active mark H3 K27 acetylation and the increased heterochromatic mark H3 K27 trimethylation. They are associated with the depletion of RNA polymerase II. So after that, we performed Hi-C. What we did, we treated cells with auxin for 14 hours, or added the top of auxin and inhibitor of RNA polymerase II, We just want to remove RNA polymerase II from chromatin as much as we can. Then finally, we FARC sorted the cells, make sure we only take G1 cells to perform Hi-C. And here, I think you probably already see from this mag-based large structure, this compartment level, there are not many differences between control and the degradation samples. But if you look within a high resolution to this loop level, you can see here the dots, clear dots are popping up everywhere. They are CDCF-based cohesion loops. And interestingly, here we can see some new loop emerge in these degradation samples, both this auxin and auxin plus inhibitor. So after analyzing all the loops, to our surprise, we found more than 800 loops only exist in degradation samples. And these loops are significantly longer than control loops. And this aggregate peak analysis say these loops have quite strong signal here. And also the loop anchor analysis say these loops have very strong insulation. So longer length than control and quite a strong signal and it's quite a strong insulation. So this say after removing POL2, there are some new CDCF-based cohesion loops are formed. And if you look within this control dividing degradation map, here red means control has more signal than degradation sample. Then blue means the degradation sample has more signal than control sample. So here you can see not only new loops are formed in degradation samples, but also new straps are formed in degradation samples. And the new straps are really say after removing POL2 of cohesion continue looping. So all of the results indicate RNA polymerase II can act as a barriers to cohesion looping in G1 phase. So after answering the first question, we think there is another very interesting time point. That is this re-enter G1 time point. Because we know one cells during mitosis, most of the protein are removed from chromatin, and chromatin is compacted a lot. But when during mitosis to G1 transition, both genome structure and gene expression need to be reorganized and reactivated. And also we know enhancer and promoter, they can form long distance interaction, they can form loops. And there is a paper from this Gerd Blober lab. They say during this mitosis to G1 transition time point, they found some enhancer and promoter loops form earlier than this CDCF-based cohesion loops. So we curious, could be RNA polymerase important there, not only for transcription, but also for this genome reorganization? To answer these questions, what we did, we took cells and we synchronized them at the check time of G2M by using an Rho 3363 inhibitor. At that time, we add auxin to degrade all the RNA polymerase II, because RNA polymerase II is not a part of mitotic chromatin. Then we release cells through mitosis and allow them to quantitatively re-enter G1. Then finally, we FARC sorted these cells to make sure we only take G1 cells to perform Hi-C. Here is Nadine did this Western blot to check the degradation. So here you can see most of the POL2 is gone, but there's still some POL2 are binding on chromatin under this re-enter G1 condition. So the first difference is what we found is only in this re-enter G1 degradation samples, we found more interactions between different chromosomes. Because I mentioned before, chromosomes are tend to occupy their own territory, and but here we found more interactions between chromosomes. So that means chromosome territory is affected by removing RNA polymerase II. And here, from this wild type and the degradation re-enter G1 degradation Hi-C map from this compartment level, here you can see this degradation map is very blurry. that means the boundary of the compartment is blurry. The separation is affected. And also we calculate and we found in this degradation sample, it has more compartment A and compartment B interactions compared than control. So the active region and inactive region is affected after removing PO2. Then if we zoom in, we look within this higher level, higher resolution, we can see here all the stripes and the interaction within a task and the boundary, all of them are become very weak. So it is a really strong effect. And if you look within the loop level, you can see a really big differences. So first, there are around 2,000 loops cannot form in degradation samples. And there are more than 2,000 loops. They are shared loop. They can form in both control and degradation. But if you see here, this shared loop in degradation samples have very weak signal. And also the loop anchor analysis say this red means the degradation sample insulation. The loops in degradation samples also have a very weak insulation. So a weak signal and weak insulation. That means something wrong with CDCF-based cohesion loops in these degradation samples. And in addition, so here we also found more than 1,000 loops only exist in our degradation samples. It's similar as the case before. But the difference is the case before. The new loops in degradation samples, they have very strong loop signal and strong insulation signal. But here in this re-enter G1 degradation, the loops have very weak signal. So that also means these CDCF-based cohesion loops have some problem. And after that, Nadim Bamasa did a fractionation Western blot. We checked the cohesion looping model relative proteins. And to our surprise, here, these two subunits of cohesion, SMC1 and RED21, they look same. And also CDCF look same, maybe a bit more in this degradation sample. But the cohesion loader, NIPBL and MOL2, and also the cohesion releaser, WAPL, are less in these degradation samples. Here is the immunofluorescence just to show cohesion are same in re-enter G1 control and re-enter G1 degradation. So this result means RNA polymerase II is associated with the loading of cohesion. So here I give a short summary is this in general, G1 RNA polymerase II can act as barriers to cohesion looping. But under re-enter G1 condition, PO2 is necessary to genome reorganization. And because all of the level like compartmentalization tasks and the loading of cohesion, all of them are affected after removing PO2. And thank you for Professor Akis Papadonis supervised this paper. And also thank you, Nadine Ubermansa did all the experiment, and thank you, Natasha, analyze the chip-seq data and the attack-seq data. I didn't show this result here because of the time. And also because of the time, I didn't show all the results of this paper. I just showed the high-C data analysis part. And thanks for all the collaborators for their great jobs. And thank you. And thank you for your attention. I'm happy to take questions.
Thank you, Shu. Thank you very much. Okay, so we'll go through questions. First question by Elia Flyermer. So have you compared degradation to transcriptional inhibition during G1 entry? So here you presented degradation results. So have you looked at transcriptional inhibition? Which
no, we just tested the degradation of RNA polymerase II. We didn't test the inhibition of transcription.
Okay. So we have a question by Roman Barth. So do you know why cohesin loaders and RNAPll is present to a less extent without RNAPll2 after mitosis?
So we are not sure about the reason now, but I mean, because we degradation the POL2 and POL2 is important for transcription and I mean, it's also indirectly important for translation. Maybe that is the reason is because of the synthesis. I mean, the protein is not enough, but maybe because I mean, there is a paper say this there is a paper about this NIPPL NIPBL chipseq. It is it's usually not usually it's like 30% of this NIPPL are localization with at this active TSS region. So we are still not sure. Not sure which is the reason of that.
Okay. So we have a question by Cedric.
Yes. Can you hear me? Yes.
Yes.
Can you hear me? Yes. Yes. Hello. Yes. I was wondering if we know something about the level of POL2 can be that is that remains on mitotic chromosomes, if there is.
Oh, sorry, I can't hear you clearly. Can you say?
So the question was, do we know something about the level of POL2 that is still bound during mitosis, the presence of POL2 during mitosis?
Thank you for your question. So you're asking if there is some how much POL2 are still at the chromatin during mitosis?
Yes.
Sorry, we don't know about it. I mean, we still have some experiments we want to do. I think we will do it. We will test it, but till now we don't know how much POL2 is still there. But I mean, because during mitosis, we know most of the protein is removed from are removed from mitosis, and RNA polymerase 2 is not the part of mitotic. But I mean, it's science. We can't say like 100% say what is what. Yeah.
Biology more than science. Yeah. So yeah, we have a question by Vipin. So where's is there any enrichment in genomic features for the regions that are looped together following RNAPll2 degradation? So is there some genomic features specific to these, new loops where you have these after degradation?
Yeah, I mean, in our result, we found this, for example, we found these new loops are longer and they are strong. I mean, maybe they are not as strong as this shared loop, but they are strong and they are, I mean, the insulation is very strong. So we think, yeah, I mean, this is a feature of these new loops. I mean, they are longer.
Yeah, but is there something specific, for example, in the sequence? Have you looked at, the sequence where you have a new loop that pops up? So have you looked at the sequences, whether they form different classes, whether there is a sequence signatures, something like that?
Sequence signature. No, I haven't. I mean, I haven't checked the motif of the sequence, the motif of the location. But I mean, I found this kind of like new loop and the new straps are actually they are like in whole genome. It's not like only on this position which I showed. I mean, they are very general. But I didn't. I haven't checked the motif of the of this location sequence. Yeah.
Okay, so I think that we are going to stop now. There are maybe a very last question by Francesca Muccianesi. Okay, so in degradation, loops, studs, and compartments are weaker. Do you observe decrease of interactions in specific ranges of distance or at all ranges with the exception of trans interactions?
No, I mean the weak, I mean, which I said after this re-enter G1, re-enter G1 degradation samples have weak everywhere. It's not like specific, it's everywhere on this whole genome and influence this TAS compartment loop every level.
Okay, actually I had a question. So from your data, it looks like actually maybe it's weaker, but the peaks look sharper. Is that right? I mean, it's more specific, meaning like it looks like more. It looks like you observe that the RNAPll hinders the action of actually these loop extrusion motors. Is that right? So
So
so so for example, we see on your slide 21, for example, um,
yeah,
21. Yes. So here, yeah, it's weaker. I
yes.
agree, but it looks sharp.
Yeah.
Sharper, which is interesting. It's have you looked at that?
I mean, yes. I mean, if we see here, but I mean, it's sharper. But you see here, they have a like a distribution of the
Yeah,
signal.
that's
I think that
that's my
might
point.
be,
So have you
yeah.
looked at really the 1D profile of these peaks? Let's say.
No, but I think
Okay.
it's a very good question. Thank you for it. I mean, yeah, thank you.